I'm happy to open the scientific program with the session Sex Differences in Autism from Animal Models to Human. And the session was organized by Sagiv Schiffman and Shlomo Wagner. Sagiv from the Department of Genetics, Hebrew University, Jerusalem, and Shlomo from the Department of Neurobiology in Haifa University. We really thank Sagiv and Shlomo for their engagement and commitment to propose and think and put together this program. And it is my pleasure to invite them to moderate this session. Thank you.
Okay, thank you very much for all this introduction. So good afternoon to everybody here. And it's a really great pleasure for me to introduce Dr. Donna Werling, who will give us the opening lecture about genomic investigation of sex differential risk for autism spectrum disorders. Dr. Werling is an assistant professor of genetics at the University of Wisconsin-Madison. And she did her PhD in neuroscience with Dan Geschwin in UCLA and a postdoc with Stefan Sanders and Matthew State in UCSF. In both places and also in her current lab, she's studying the mechanism underlying sex differences in autism. So it's really great that she had time for us and to share with us her work on this subject. Thank you very much, and the stage is yours.
Thank you so much for that introduction, Professor Schiffman, and thank you for inviting me to present today. Great, you can see my slides now. So I am really excited to be here, to be kicking off this session on sex differences in autism. So we'll dive right in. We're going to be talking about genomic approaches to approaching this question. Now, I know many folks in the room may not need this introduction, so I'll go quickly, but simply autism spectrum disorders are pervasive neurodevelopmental disorders that are characterized by the co-occurrence of two symptom domains in the same individual: challenges with social communication and the presence of restricted or repetitive behaviors or interests. Now, the prevalence of autism spectrum disorders is approaching 2%, at least in the United States, where the stats on this slide come from. And autism is an incredibly heterogeneously presenting condition that is characterized by a wide range of abilities and disabilities within the population of folks who have an autism diagnosis. And this is maybe most simply illustrated by looking at the distribution of IQ among autistic patients. So approximately 1/3 of individuals with an autism diagnosis have an IQ in the intellectual disability range, whereas upwards of 40% of patients have an IQ in the average to above average range. I think this nicely illustrates the wide range of abilities and disabilities under this spectrum. And we know that genetic variants are associated with autism risk. Now, the aspect of autism that is motivating much of our work who are attending this session and presenting here and are motivating this session in the first place is that autism is consistently diagnosed about 4 times more frequently in males than in females. And what you're seeing in these bar plots are prevalence estimates in males in blue and females in red. And what we see is that this male bias in diagnoses is consistent over time and over place, whether you're looking at monitoring sites within the US, as this plot shows, or whether you're looking across countries. If we convert these prevalence estimates to a sex ratio of cases, we see that that no matter the year, no matter the location, the difference in prevalence between males and females comes out to about a 4-fold higher rate of diagnoses in males. Now, a logical question to ask then is whether autism looks the same in males and females, or if there are any differences in the way that males and females tend to present their autistic symptoms. And this is not necessarily my area of expertise, but I will draw from the literature, which is growing. Research in this area from clinical and behavioral research groups are really tackling this topic. And this is a very active area of research, but a few patterns from this work start to stand out. The first is that overall, among people who have an autism diagnosis, males and females tend to have comparable severity of symptoms on average, at least to the extent that we can quantify autism severity. But multiple studies have also reported that diagnosed females may have reduced scores on measures of restricted interests and repetitive behaviors as compared to males. Diagnosed females are also more likely to have an IQ in the intellectual disability range. They're more likely to use what we call camouflaging, or an overt performance of social routines to mask social challenges, and they may be more likely to show co-occurring internalizing disorders like depression and anxiety. But regarding the reduced, restricted, and repetitive behaviors in females, it may be the case that females' restricted interests are focused on topics that are less often recognized, and that updating our diagnostic measures to include more exemplars of female typical symptoms could help alleviate this difference. Regarding lower IQ, this may be due to ascertainment into studies. Females may need to present with additional challenges, like intellectual disability, in order to come to the attention of their teachers or doctors and be evaluated for a diagnosis. Camouflaging behavior is a very important phenomenon to understand, and there are research groups actively working on this. Right now, it appears this is a challenging phenomenon to quantify, but watch this space for more research in the future. And finally, non-autistic adolescent females are also more likely than males to have co-occurring internalizing disorders. So this may not be a pattern that is specific to autism. So in short, there are some dimensions of autism symptoms and co-occurring features that may be different between the sexes, but there is no phenotypic feature of autism that is as striking or as consistent as its male bias prevalence. But we should keep in mind that these patterns refer only to sex differences among the group of people who have an autism diagnosis. So as it stands, the current diagnostic symptom may actually minimize sex effects because the autistic females who are phenotypically most similar to males may be the ones who are more likely to receive a diagnosis. So these patterns say very little about symptoms in folks who go unrecognized and undiagnosed. So it's a complex question. Now, one working hypothesis to explain the male bias in autism's prevalence is that there may be protective mechanisms in females that are reducing their risk for autism, or vulnerability mechanisms operating in males that may put males at greater risk for autism. In other words, that there's something about females' biology that reduces the impact of other autism risk factors, such that fewer females wind up presenting with autism, or there's something about males' biology that amplifies the impact of those risk factors, such that more males wind up presenting with autism. And these are not mutually exclusive possibilities. Do we have any biological evidence so far to support the existence of female protection or male risk? Yes, and to understand this, I would like to introduce the concept of the female protective effect model. So we start with this liability threshold model, where we're assuming that liability or risk factors for autism are normally distributed in a population, and that there is a minimum level of liability or a threshold beyond which, if an individual has experienced this high level of liability, they'll wind up presenting with a diagnosable phenotype. Now, in the case of autism, where we have striking sex difference in prevalence, we have to modify this model, and we now claim that males and females have different thresholds, different levels of minimum liability that they need to meet before they present with an autistic phenotype. So in other words here, females, we assume, are able to withstand a higher level of liability than males before they cross that threshold and present with an autistic phenotype. Now, this seems logical, but do data support this model? We can test this by taking a close look at this upper tail of the distribution outlined in green. So let's zoom in. Now, one major prediction from this model is that autistic females should have a greater burden of risk factors than males. So I'll remind you that this is a quantitative axis. We're moving, move to the right, we are increasing in liability. And so if we're assuming there are two different thresholds in the sexes, if you're looking at a population of folks who have an autism diagnosis, the average liability experienced by females is higher on the tail and therefore likely to be greater than the mean liability experienced by your population of males. Now, the dimension of autism risk that we understand the best currently is genetics. So let's rephrase this to make a more specific prediction, which is that we predict from this model that autistic females should have a greater genetic burden than diagnosed autistic males. And genetic studies do support this prediction. So it turns out that the incidence of deleterious de novo variants is higher in autistic females than autistic males. And this is true whether you're looking at de novo copy number variants, higher incidence in females, whether you're looking at de novo indels or insertion deletion variants, also higher in females, or whether you're looking at de novo protein truncating variants in highly constrained genes, the incidence of these types of variants is higher in females than in males. And the consistency of this pattern across these three independent classes of genetic variants that are all themselves associated with autism risk supports this prediction from the female protective effect model and suggests that a female protective or male vulnerability effect is likely to exist. So we're brought back to this key question then. What is the mechanism involved in protecting females or in amplifying males' vulnerability? Or at least to start, what are the players involved in such a mechanism? Genomic approaches can provide a useful tool for discovering the potential players involved in these mechanisms. So thanks to genomic technology, next generation sequencing, today analyses of DNA and RNA can be run at a genomic scale, testing every gene in the same analysis. So imagine this network image represents all of the genes in the genome. Genomic analyses are said to be unbiased in that you can test this whole genome at once and without a priori hypotheses about the biological functions that are involved, you can let the statistically significant observations from your data highlight the parts of the genome or the transcriptome that are changed in your group of interest. So genetics and functional genomics studies both share this unbiased framework. And we'll touch on what genetics have shown us so far about sex effects in autism, and then we'll spend the remainder of the time thinking about transcriptomics. So a logical early hypothesis about autism risk was that it may be an X-linked condition, which makes sense given its male bias prevalence. But while some syndromic forms of autism like Fragile X and Rett syndrome have been linked to X chromosome genes, the vast majority of the risk loci found to be associated with autism today are on the autosomes. For example, these 102 risk genes here were discovered by exome sequencing to find de novo and rare variants of large effect in these genes. And so these are all autosomal. On the one hand, this rate of gene discovery on the autosomes shows us that the X chromosome alone is not likely to fully account for the male bias in autism. But on the other hand, risk genes have predominantly been found on the autosomes because genetic data on the X chromosome are somewhat uniquely underpowered in autism. And this is due to two things. One, that autism is a male bias condition, so the majority of autistic cohorts that exist out there have predominantly male cases, which makes sense, combined with the fact that the majority of this gene discovery is driven by looking at de novo variation. And about 80% of de novo point mutations occur on the paternal lineage during spermatogenesis. So when you're looking at an autism cohort where you have a majority of male cases, all of those males do not inherit their paternal X chromosome. They only get a maternal X chromosome, which has only about 20% of the de novo point mutations that one could potentially occur. And so this leaves us with the observation of fewer de novo variants in genes on the X chromosome in these cohorts with which to discover risk genes. So interesting to think about. But what these data together show is that autism is clearly not an exclusively X-linked trait, though larger samples in the future may yet be able to attribute a larger fraction of autism sex bias to genes on the X. We will see. Beyond the X chromosome, it's possible that certain autosomal risk loci could still confer sex-biased risk. For example, if one of these genes is the target of a sex hormone receptor regulation, for example. Now, to find these sorts of genes, Tishel Turner and colleagues ran a study on genetic data from two large cohorts of a combination of autism and neurodevelopmental disorder cases. So the results on this slide are not specific to autism, but I think it's a interesting study to point out nonetheless. And this analysis found that there are a number of genes that meet genome-wide significance for association only in one sex or the other. So in females in yellow and males in blue here, which is really worth noting. There really may be these individual genes that themselves are sex differentially regulated and associated with risk. That said, only a small number of these genes replicate the sex-biased effect across the two cohorts that these researchers looked at. But this sort of approach could be used to identify specific genes that may have sex differential effects. And these genes would be excellent starting points for neurobiology work to learn about the mechanisms driving these apparent sex differential effects. So this is a snapshot of the status of genomic studies looking at sex differences and represents where we currently stand there. Which brings us to the next question. What can transcriptomic studies tell us about mechanisms involved in sex-biased risk for autism? And for this topic, we'll walk it back a little bit and then move up to present day. So 5 years ago now, I published a study that aimed to use gene expression data to ask a simple question about how sex differential biology interacts with autism etiology. Specifically, we were asking, are autism risk genes themselves sex differentially regulated? In other words, are these risk genes direct targets of sex differential regulation, whether by sex hormone receptor binding or transcription factors from the X or Y chromosome, for example? Or do sex effects intersect with autism etiological pathways somewhere downstream from the risk genes themselves? And the goal of this work was to try to find a trace of a risk modulating mechanism that could point us toward a pathway or a process that might lead to increased male vulnerability or decreased female vulnerability. And the approach was simple. We used transcriptome data from neurotypical brain tissue, largely from the BrainSpan resource, to characterize sex differential expression in the typical brain, and then we compared these sex differences to gene sets that had been implicated in autism biology, whether that be autism risk genes identified by exome sequencing, for example, or genes that have dysregulated expression in the autistic brain as identified by transcriptomic studies from brain tissue. Now, it needs to be pointed out that this analysis at the time was based on cortex tissue samples from a small number of neurotypical donors. So the analysis was run in 3 sets of data, each of which had cortex samples from no more than 5 unique male and 5 unique female donors. And at the time I was working on this, we also had a much smaller number of high confidence autism risk genes and just 2 major studies of gene expression differences in the autistic brain. But nonetheless, we had tested these small adult and prenatal sample sets for transcriptome-wide sex differential expression, and then asked the questions, are autism risk genes sex differentially expressed? And if not, what is? And can any of those differences be linked to autism pathology? And we found that autism risk genes are not sex differentially expressed. Each of these lists in these columns corresponds to a set of genes associated with autism risk by virtue of genetic variation, and we see no preferential enrichment of these gene sets with male biased or female biased genes. We did see, however, that gene sets that were upregulated in the autistic brain and associated with astrocyte, microglial, or immune function are also elevated in neurotypical males as compared to females. And we do see this weaker, but still significant, enrichment of female-biased expression for genes downregulated in autism and associated with neuron function. So this pattern suggested that sex effects on autism etiology are likely or more likely to be acting downstream from autism risk genes. If I can illustrate this result in a slightly different way, here are the genes in a module, a neuron-associated module identified by comparing autistic to control brain. These genes have decreased expression in autism. And what this plot is showing is the fold change for the autism versus control comparison in orange and the male versus female comparison in blue. And by and large, the direction of change agrees. These genes are reduced in autism, and the majority of these genes are also reduced in neurotypical males. For this M16 module, we saw the reverse. So M16 module genes are these immune function related glial genes that are elevated in the autistic brain. And again, the direction of effect largely agrees between the autism and sex comparison. These genes are elevated in autism in orange, and the majority are also elevated in neurotypical males compared with females. Importantly, these autism-associated expression changes that show these parallel responses in our sex comparison, these autism changes are not themselves male specific. So what we're looking at here is the first principal component of gene expression for these M12 genes and M16 genes in the study of autistic versus control data. And what you see is these M12 genes tend to be downregulated in autism compared with controls, and these M16 genes tend to be upregulated in autism versus controls. And I've marked off the female samples with the black bars in both sets. So what this shows us is that tissue from autistic females is just as likely to show these expression changes of these two sets of genes as is tissue from autistic males. So we think these patterns are capturing autism state phenomena that are not confounded with sex, which is reassuring. Now, in addition to looking at gene sets with altered expression in the autistic cortex, we also tested whether sex differentially expressed genes were enriched for genes that mark the function of specific cell types. And here we find patterns consistent with the interpretation of that M16 module function, in that male biased genes are enriched for markers of astrocyte and microglial cells. These findings suggested to us that sex effects and autism neurobiology intersect, number one, downstream from autism risk genes, and two, that this intersection may involve the function of microglia and astrocytes. Specifically, it looks like neurotypical males' gene expression with regard to glial and immune processes looks more similar to the autistic brain than neurotypical females. Translating these expression patterns to that liability threshold model, it could be that males' baseline glial biology puts them one step closer to an autistic phenotype than females, and that this shift in the mean distribution of liability could be sufficient to lead to, or at least contribute to, a 4-to-1 male skew at the tail of the distribution and a fourfold increased risk for autism in males. Now, this is all very interesting, but given just how few individuals contributed to the samples that brought us to this conclusion, and given how far discovery of risk genes has come for autism, these results are certainly due for a recheck, due to be validated. And my lab has started exploring this in a data set called BrainVar, which was published on last year. This consists of paired whole genome sequencing and RNA sequencing from dorsolateral prefrontal cortex tissue from 176 unique human donors that range in age from 6 post-conception weeks through 20 years of age. We've been focusing on 2 stages of development where the male and female samples span similar age ranges, a prenatal stage and a postnatal stage. Lee Kissel, a graduate student in my lab, ran sex differential expression analysis in the RNA-seq data in both stages and tested similar hypotheses as before, but now with updated gene lists. Now, taking a global look at our results shows that, like many previous analyses of sex differential expression, other than the Y chromosome in blue and the exist transcript and other X chromosome genes in red, sex differences in expression are small in magnitude and not terribly significant, which you can see in these autosomal-only plots here. So despite our larger sample size, discovery of robust, individually significant sex differentially expressed genes is still sparse. These effects are small, both prenatally and postnatally. And yet, if we look at how specific gene sets behave, we do see some skews. For example, this set of genes from now a more recent study of gene expression in the autistic brain, called the M19 module, has elevated expression in autism, and again, it's associated with microglial and immune functions. So genes in this set tend to have male skewed expression, especially prenatally. These genes lie on the right side of these volcano plots. Now, small magnitude shifts like these may very well be noise, but it's also possible that subtle shifts in expression across a functionally related set of genes could indicate that the pathway or process that these genes represent is collectively upregulated or downregulated in one sex or the other. And our main interest here was not necessarily to identify genome-wide significant sex differentially expressed genes, although that would be fantastic, but to try to replicate the earlier findings that I had just shared with you that had come from those much smaller samples. So we decided to follow the method used in the 2016 paper and apply these minimally stringent thresholds for identifying sets of sex differentially expressed genes, just a simple p-value of 0.05 and a minimum fold change magnitude of 1.2. We then tested these sex differentially expressed gene sets for enrichment with autism-associated genes. First up, we compared them to these now 102 autism risk genes from exome sequencing. And I would show you a plot for this, but there's nothing to show. 0 out of the 102 known risk genes are sex differentially expressed, even at these more generous thresholds. Next, we looked at enrichment for these 6 autism-associated co-expression modules that were identified from the most recent study of transcriptomics in the autistic brain coming out of the Geschwin lab. And purple spots in this heat map are depletion or less overlap than expected between gene sets. Green spots are enrichment or more overlap than expected. And the only comparison that is significant here after correction for multiple testing is the enrichment of prenatal male bias genes for this M19 module. And this result agrees with the previously reported patterns, this enrichment in male bias genes for glia-associated autism upregulated gene sets. And critically, this result is not dependent on the brainspan samples that were also tested in our previous work. When we exclude samples from the brainspan cohort and use only those samples that were unique to BrainVar, we see the same pattern. Now, this validation gives us more confidence that this overlap between neurotypical male and autistic biology is real, while at the same time, we are aware that the sex biases driving this result are very small in magnitude, which does challenge our interpretation of this finding somewhat. But this is where we stand today, and time is running short, so I will summarize here a few of the things that we've talked about, which is that we know that autism has dramatically male bias prevalence, but more nuanced sex differences in its phenotype. Genetic studies to date have not yet identified very many sex differential risk loci, although that list may grow as sample sizes grow. Transcriptome analyses can be used to identify the potential players involved in these sex differential risk mechanisms, and the analyses I've shared with you today seem to be pointing toward genes associated with immune and glial function as being kind of parallel, upregulated in parallel fashion in autism and neurotypical males. Now, there are many more questions that remain, but a few that are sparked by these analyses I've listed here. So regarding these immune and glial genes, are these differences by sex and in autism driven by changes in cell type composition or the function of these cell types or processes? And beyond that, what is the mechanism? How might increased immune and glial activity alter brain function and increase the likelihood of presenting an autistic phenotype? And the second questions really hit on this small magnitude effect size of these sex differences in the frontal cortex. The mid-fetal prefrontal cortex has really been honed in on as a site for autism pathology. Autism risk genes are strongly expressed and co-expressed here, but it may not be the right spot to look for strong sex effects that alter those phenotypes. Should we be looking in other brain regions or only in specific cell types or even at different ages? So there's much work left to do, and I have to say I'm very much looking forward to hearing the other talks in this session to learn more about how others are addressing these unknowns. And with that, I will thank my lab members and collaborators and funding sources, and thank you all for your attention.
Thank you very, very much for this wonderful talk. If any in the audience have questions, you're welcome to post it in the chat. I have already here one question that was posted. I'll try to translate it. My question to the first speaker, if we can indicate on any impacts of hard socio-economic situation on development of symptoms of autism among children?
That's a really important question. and is probably beyond my area of expertise. But I know that there are groups around the country, maybe I know the Marcus Autism Center at Emory, I heard Ami Klin is speaking tomorrow, has some studies in this regard looking into this. But yeah, I don't know that I can speak to this question in a comprehensive way. It's almost certainly likely to impact, if not the development of these individuals directly, certainly their access to care and the likelihood that one would get a diagnosis.
Yeah. So there is a worry here that there is a bias in diagnosis because of also the misdiagnosis and autism girls. There is another question here that somebody asked about whether you can quantify how much of the sex bias in autism can be explained by the transcription differences that you see.
Again, a really great question. I think that we cannot quantify that right now. And the reason for that is because the bulk of these transcriptional differences come from post-mortem human brain tissue. So that BrainVar data set has a fairly large sample of individuals, but when we're trying to see how the sex differential patterns map onto autism risk, we need to look at autistic brain tissue, and the samples that are available currently, the sample sizes that are available to look specifically at autistic brain tissue are quite small. I actually put out a small review about this wrote this in the fall, looking at how many individual donors have contributed to these autism versus control transcriptomic brain data sets so far. And in terms of things that have been published, there are only 14 autistic females who have been included in these studies. So that right there is a really severe limitation on us being able to look at how these expression patterns differ between autistic males and females themselves, and then be able to extrapolate that to wider risk. So hopefully, I mean, there are efforts underway to increase those sample collections, and hopefully data sets will grow. But because the data is based on brain and not something easily collected like blood, it makes it challenging to estimate that.
Great, thank you very, very much.